to remind you that if you come here at this time next week, you will feel very alone. Because next week, instead of having the physics colloquium at our normal time, we'll have the Segre lecture, which will be in Zellerbach Hall at 6 p.m. So this will be the lecture delivered by Ray Weiss. So we'll hear all about beginning the exploration of the universe with gravitational waves. And so I hope to see many of you there for that. But on to our feature attraction for today. So Sarah Keller has been a professor in chemistry and physics at the University of Washington since 2000, although she's fairly familiar to a number of us here at Berkeley, especially since not even a year ago, she was a visiting Miller professor. Sarah received her PhD at Princeton and did postdocs at UCB, or sorry, not UCB, that's here, UCSB, right? And Stanford with two different prestigious postdoctoral fellowships. She's certainly no stranger to many of us here. I often hold her research that she says is how simple lipid mixtures within bilayer membranes give rise to complex behavior. I hold that up as a really beautiful example of the universality of critical phenomena, as I'm sure you'll see here today. I also hold up Sarah personally as a sort of singular example of a scientist who, as you can see, is alive and well. And well, most of the ones we talk about in class aren't anymore. And most of them are also not women. And so I think that this is particularly important to have examples of people who are doing important science full of life that we can talk about in the context of the work that we're teaching people in their classes. So I feel a little bit bad pointing out that Sarah's a woman, although that's probably somewhat clear. She is first and foremost a scientist, not only a female scientist, but I hope that in so doing I can show our students that role models can also be diverse. And she indeed is a stellar role model. She's even won awards for this. So she received the UW Postdoc Association Mentor Award a few years ago, in addition to some departmental and even university-wide teaching awards. Scientifically, her accomplishments have also garnered many awards, ranging from the Biophysical Society's Tom Thomson Award and Dayhoff Award, a Cottrell, and a career. She's also a fellow of APS and AAAS. So thanks for joining us again, and I'm looking forward to a lively seminar.
The mic working? Okay, excellent. It's such a pleasure to be back here. There's so many friends in the audience. It's really nice to see you all. So that aspect of seeing so many friends, and that I was here for an entire month in January, and I was somewhat hoodwinked into giving 4 different talks while I was here in January, left me with this challenge. What will I possibly present to you, my friends, who might have seen so many of my previous talks? So luckily David Chandler could not be here because he was the one who I know had seen all 4 of them. So he's out of town today. So here's my challenge, is that so many of you are familiar and know my work. How will I engage you, as well as the new friends that I have yet to make? This is a joint MCB and physics seminar. How will I engage both of those communities? And some of you are experimentalists and some of you are theorists. How am I going to engage all of you? So maybe you can give me some feedback at the end of this if I have managed to do that. And what I did not also realize is yet another challenge is that I have visited you on the hottest day of the autumn, and we are all already very warm and wishing to be out in the breeze. And how will I manage to keep all of you awake in such a warm room? So there we go. I titled this today's fresh sheet because with all of those challenges, and for those of you who've seen my work before, I am going to try to limit myself telling you results that were published only in the past year, with the caveat that I do need to introduce the system to you. So I also know that there are a good population of physicists here. Physicists typically are not so shy about interrupting, and I'm assuming that will be the case here, and I welcome those interruptions throughout. Okay, here we go. My lab studies giant unilamellar vesicles. So these are single-walled sheets of lipid bilayers. They are curved into a thin shell, and when they are made of a particular lipid composition, so these require only 3 different lipid types, they phase separate into 2 coexisting liquid phases. And I'm really sorry, they have kind of silly names, and I didn't make them up. The 2 liquid phases are called the liquid disordered phase and the liquid ordered phase. And this is just a schematic of the systems. And what I would like to tell you as the place that we are going to end up here, some of those new results, is to notice that in this system, we only ever see a dark domain and a bright domain. We never see one of an intermediate value. So in other words, we never see an incomplete overlap like this of the domains. The 2 domains that we see, well, how bright and dark are they, are going to depend on the extent of the phase separation in the system, the composition of the 2 phases. So when we look in our microscope, we've added a dye that prefers to be in one of those 2 phases. We'll either see white versus black, and that says the composition of the 2 phases are very different from one another. Or we'll see a light gray versus a dark gray. For the physicists in the audience, that means that the tie lines in that system are shorter. That system is approaching a critical point, and that's the research that Naomi alluded to before, but I'm going to disappoint you and not talk about critical phenomena because I'm only going to talk about results published in the last year. But I'd be happy to tell you at length about it because I think it's so beautiful after the talk. Okay. Now, what may be obvious to the MCB folks in the audience and maybe not so obvious to the physicists is that there is a biological motivation for thinking about domains and membranes, and that comes from thinking about the outer membrane of a cell that is called the plasma membrane. If we think about only that membrane and not the internal membranes, then that membrane is thought to be not uniform and to make regions that are enriched in particular lipid and protein types. In the literature, this is called rafts, or there are a bunch of different names for it now as that term is going out of favor in the community. But that's a motivation in our system. Concomitantly, what may be obvious to the physicists in the audience, and maybe not so much to the MCB people, is that when we look at a system like this, and when we're talking about coexisting phases, we're necessarily talking about a phase transition. At high temperature, entropy always wins. And so at high temperature, these systems mix into one uniform phase. Here we are seeing a vesicle. It's one big gray, exciting blob. The outline that you see here is the actual diameter of the vesicle. It's enormous. And the movie that you're going to see involves a temperature quench. That quench will nucleate small circular domains, and then the temperature is going to be constant through time, and we're going to see those domains coarsen. That kind of behavior is really universal in that physics sense of the word. And when we look at critical phenomena in this system, it is also universal. It belongs to the universality class of the 2D Ising model. So here we have a quench. Down from high temperature, small circular domains nucleate. They are running around on the surface of that vesicle. When 2 domains collide, if you watch these ones for a little bit, boop, they coalesce. And this will happen throughout time. So if we had a lot of patients and we watched this movie for maybe 15 minutes, we would see that eventually we would end up with only one bright domain and one dark domain. You good so far? No physicists have interrupted yet? Ah!
You said earlier that you never get overlapping. So at the time I thought you meant, oh, you never get a situation where you have a bunch of little domains hanging out together and giving you something in between light gray and dark gray. You must mean something other than that, because here you start from that and then they coalesce. Right.
That's a great question. I don't know if you could hear in the back, so let me repeat the question. I think to put it in a nutshell, is what exactly do you mean that you never see an incomplete overlap of light gray and dark gray? So here we see 2 fluorescence levels. There's a bright and there's a dark. It is the case that 2 dark domains can come together and then make a larger dark domain. But it is not the case that the gray levels will be, say, 1 and 1, and then they'll make 2. So that must mean that in this is a bilayer membrane. It must mean that the two phases are in registration across both leaflets of the membrane. And that's especially what I'm going to talk about later, is that the magnitude of that coupling. I thought I saw another question as well. Is that true? No, there was a head scratcher or something. Okay. Okay. There are 3 lipid types in the particular lipid vesicles that we use. Now, what I would like to say for both the physicists and the MCD community, a joy is that it really does not matter so much which particular lipids you use. So if you have particular favorite lipids, I would like to invite you to think of all of these results in the context of the lipids that mean the most to you. And for the physicists, I would like to say, don't worry about it. It's okay. It's all universal. So the 3 lipid types that we're going to use are the blue guys, the green guys, and the orange guys for the physicists. What do those mean? Those are high melting temperature lipids, they're low melting temperature lipids, and there's some sterol that is probably something that looks like cholesterol. What do I mean by high and low? This is just a relative term of those two to each other. But again, don't run for the doors because you see some chemical structures. This is a universal thing. And it doesn't matter if we start adding in more components, we will still see the same behavior. Okay, now, which ratio of those 3 components might be best to use in your experiment? Now, this is a question that is especially pertinent, I think, for the MCB community, because what is known from a physics community is how to think about phase diagrams. So, that means I need to introduce you to phase diagrams. So here it will go really fast. For the physicists who already know this, I invite you to take a 2-minute break, and then we'll come back. Here we go. We're going to think about how we might represent all the different ratios of 3 different components. In this case, let's think about colors rather than particular lipids. So here is 100% magenta. Here is 100% yellow. Here's 100% cyan. And if we would like to make, say, a binary mixture of 2 of those colors, then it will appear on one of those axes. So going from cyan to yellow, the middle of that, well, that's green. It's intuitive. If we'd like to make a tan color, tan is mostly yellow. So in fact, this one happens to be 50% yellow. And we've added a little bit of magenta, 30% magenta, and 20% cyan. So that says that every point on that triangle represents some ratio, different ratio of the 3 different components. Here we'd like to think instead about 3 different components of 3 different lipids. This one happens to be the high melting, this one happens to be the low melting, and there's cholesterol. And this work was all done by Joan Bleecker, who's now a lecturer at UW-Tacoma. What Joan did was thought about, can she find different ratios of those different lipids that will all give the same mixing temperature? What does that mean? At these temperatures, that vesicle is in one uniform phase. At these low temperatures, the vesicle has polka dots, 2 coexisting phases. And what's striking is that these 4 different compositions give all the same mixing temperature, but different mismatches of their thickness. So that's saying that the dark regions of that membrane are much thicker over here than the bright domains. And if they're much thicker versus thin, if there's a bigger difference in the thicknesses, that says that there's a bigger contrast between the 2 phases. So if we were to look at this by fluorescence microscopy, over here we would see really black domains on a white background. And over here we would see just light gray and dark gray. And this is really helpful to know. This is a lot of what we've done over the past 15 years, is to map out phase diagrams. Why is that good to know? Because now when you're doing experiments, it would be very nice to change only one variable. And so this is a way that you can be clever about choosing the lipid compositions so that you're thinking about only one variable. Maybe you'd like to do an experiment in which you only think about the thickness mismatch, Or maybe you'd like to do an experiment in which you think about only changing that miscibility temperature and keeping the thickness mismatch the same. That happens when we walk along what's called a tie line. The two end points of a tie line in a phase diagram say what the composition of the two phases is. So in every picture here, the ratio of lipids in the liquid ordered phase is the same for every system, and for the liquid disordered, it's the same. Good? All right. How do we make the vesicles? All right. So the last one was something that might be obvious to the physicists, but not so much to the MCB folks. Here's something that might be obvious to the experimentalists, but not so obvious to the theorists, is that it really matters how we make the vesicles. Our laboratory makes vesicles by electroformation, which is also a favored system to use in many other systems here at Berkeley. Another way to use it is to make those vesicles is by sea dice. And this is work done by Ben Horst, who is sitting right up there. And he's now a grad student here at Berkeley. So what I'd like to tell you is that it is difficult to make liquid phases of known composition by this new method. This new method, very briefly, is an acronym for a very long name invented by these people called continuous droplet interface crossing encapsulation. Here's how it goes. We have a little spinning dish. It's a petri dish. On the inside, there's a cylinder of air. And then there are layers of different liquids. So here is a layer of decane. Here is a layer of heavy mineral oil that contains lipids. Here is a layer of aqueous solution. And the technology here is to insert a pipette and have water droplets that are sheared off by the spinning liquid as it goes around through the decane, through the heavy mineral oil, into this aqueous solution. And they make really quite beautiful vesicles. And these are Ben and Matt's really pretty pictures. So there's a nice uniform vesicle. This is achievable. So this is prior art. What we were particularly interested in is, can we use this same technique to make the kinds of lipid compositions that we care about in our system? And I'm sorry to say, not so much. There are two pathologies that persist in this system. The first one is that we often see inclusions of oil. Those show up as bright regions. And the other is that those domains, oh, they look kind of different, don't they? They're not circular. So that says that we don't have two coexisting liquid phases. Not seeing two coexisting liquid phases implies that our system does not have enough sterol. And that's crazy, because look how much sterol Ben and Matt put into the system, 100 times more sterol than anything else. So the sterol is not getting into the membranes. And so for those of you who are looking for new ways of making giant vesicles, this is a caveat to say this might not be your favorite way to do it. Good so far? It's very warm, isn't it? Okay, now to the main story here. Domains in both membrane leaflets line up. So what I'm showing in just this cartoon is that here's a domain of a liquid ordered region, and it's right across from a domain of another liquid ordered region. That implies that there's transbilayer coupling. Now the question is that coupling strong or is it weak? There was a really nice, before we started thinking about this, there was a really lovely paper by Philippe Deveau, which can be summarized as, oh, come on, you've got olive oil. How are these things possibly talking to each other across the two leaflets of a bilayer? Surely it must be the case that in order to have coupling, you need to have a transmembrane protein. And it was a pretty compelling argument. And it turned out that he was incorrect. So the evidence that the coupling is strong, we have two different lines of evidence. One is some old data, and I promised I'm not going to show you any data that's less than a year old. So I'd be pleased to show you that after the talk. And that is qualitative results in which we made asymmetric membranes, and we looked at the domain formation in each of those cases. Instead, here, I'm going to show you quantitative results from 2015 with symmetric membranes. Good so far? Okay. Here we go. Quantifying the strength of the transbilayer coupling between domains in each leaflet of a lipid bilayer. I especially want to give credit for this idea to Aurelia Honerkamp-Smith. So the complete idea of how to go about doing this experiment was from Aurelia. Aurelia was a graduate student in my group and then went on to do a postdoc in the University of Cambridge, and we met again at the Biophysical Society meeting over some very good Thai food and a lot of beer. And Aurelia said, I just read this really fantastic, interesting thesis, and here is how you should do the experiment. I don't have time to do it on my own. And Matt and I looked at each other and went, that is how we should do the experiment. And so Aurelia's idea, all of the implementation came from Matt. Now, we're going to get experimental results, and then we're going to tie it into the theory. And the theory here is Mikko Hataya, who's at the Princeton University, and his graduate student, Tao Han. Okay. So I'm trying to convince you, we never see an incomplete overlap of domains. We never see domains travel seemingly across each other and then past each other. That must mean that there is some energy penalty for that to happen. And we're going to call that an energy penalty to misregister domains. And that's going to come in terms of a free energy cost per unit area. Now, the mere fact that we don't see domains like that puts a very low limit in terms of what that energy penalty should be. So, right, we're doing microscopy. So we can only see things that are micron scale. So that misregistration that we would see would have to be in terms of microns squared. That observation puts this incredibly weak limit on the interleaflet coupling parameter. That interleaflet coupling parameter is the free energy cost per unit area to misregister a domain. Now, there were various forms of simulations and predictions in the literature about what that value should be, but there'd been no previous measurement. And so that's where we came in, really wanting to know what this number was. Our goal is to drive the domains. Like, those domains don't go out of registration on their own. We need to drive the domains out of registration. And Aurelia's huge contribution to this is that she had recently read Peter Janssen's thesis, in which he'd done some beautiful experiments, putting bilayers down on surfaces in microfluidic channels and then driving them forward. So in other words, this is called tank treading. And the idea is, here we have our substrate, and there are the two different leaflets of our bilayer to make our membrane. We're going to flow water past on top in a constrained channel, in a microfluidic channel. And that is going to push the upper leaflet of that bilayer with respect to the bottom leaflet. So the bottom stuck. It is not going to move. And the top leaflet, being pushed by the coupling with the water, is going to travel on top of it and lay itself down as it travels. Good? Okay. We can recapitulate Peter's work. So looking at this system now from the top view, okay? So what we're looking at here is the bright bit, and there's some bare glass, some dark bare glass behind it. And as we push, we can see the front of that lipid bilayer as it lays down new material. Aurelia's inspiration was to think, instead of doing that with a uniform bilayer that has the same lipid composition everywhere, what if we now do that with a system that has domains? So her idea is to take a bilayer that has domains, push on the top, and then misregister the top with respect to the bottom, so that looking from the side, you'll have domains that have a region here that is misregistered. Okay, now that's where we left from the Biophysical Society, and then it was Matt's job to figure out how to actually implement that in real life, which turned out to be pretty challenging. The way that he did that was first to make giant vesicles that have micron scale domains, bring them down on a substrate, then burst those domains down on the substrate, So now we have a bilayer that has domains. Next, we need to backfill with vesicles that have the same lipid composition as the liquid disordered phase. So that means that there's going to be no net transfer of lipid composition from the bright areas from the splatted vesicle to these surrounding areas. And the reason that we can do that is because we can build on our decade of previous experience of mapping phase diagrams and mapping tie lines, so we know exactly what the compositions of the two different phases are. All right, then apply flow in a microfluidic channel and measure. So this is the exciting bit for us. This is a threshold hydrodynamic shear stress, and this is an inverse size of domains. Now, you already know the answer that if there is no shear stress down here, then those domains must be stationary. They're not moving. And if we push hard enough, then we should get those domains to move. What's slightly counterintuitive to me is the idea that this is an inverse size of domain. So these are bigger domains. You have to push less in order to get the big domains to move than the small domains. Small domains are harder to get to move. Okay. Now from this we can take the slope and we get a value of the hydrodynamic shear stress versus the inverse size of domains. Now that's really exciting for me as an experimentalist, completely uninteresting to the theorists who had made those predictions, because they want to know what is the value of that interleaflet coupling parameter? What is, how much energy per unit area do you need to misregister domains? And so that's where we teamed up with Mikko Hataya. So we need to go from these data to a value of the interleaflet coupling parameter, and here's how we do it. There are 4 forces in the system. 2 of them are driving forces. So let me set up so that flow is going in this direction. So the first force that is the driving force that's going to make domains go in a particular direction is the shear. It's the water flowing on top. And that's because that water is coupled to the head groups of the lipids. The next one is due to drag. What's happening is that as the water is coupling to those lipids, it is dragging lipid around a domain. And so that's going to make the domain go forward. And we know this, as scientists, we have so many friends that when we go to a concert, we're just getting dragged by our huge crowd of friends forward, and that's pushing us along with them. Now, there are 2 restoring forces. The first one is the frictional force, and that's how much the individual bilayer is coupling down below, no matter what the composition is. And what we really care about is a registration force. That says that a particular domain would like to be opposite a domain in the other region. And even though there are these forces driving it forward, it's going to stay stationary. All of those come with equations. Here is the shear. So that makes sense. These are the 2 driving forces. They depend on the shear. You'll notice that the dimensionality of the length scale changes. This one goes like area, this one goes like area, this one goes like area, this one goes like length. Why? Because the way that we're going to do our experiment is to identify the very first moment at which those domains are no longer in exact registration, when they're very first slightly out, not when they're totally apart, but the very first moment. So our experiment back here, oops, other way. Our experiment back here is making steps in increasing the hydrodynamic shear stress. So here, everything at this particular size was stationary. Boom, at this one it moves. And deciding whether it moves is just whether it first comes out of registration. Okay. That gives us this beautiful equation. In fact, I should give credit where credit is due. That gives Tao and Mikko this beautiful equation. Why is this so beautiful? Beautiful in this really physics sense, right? Beautiful because this is a single parameter fit. We know, or we can measure everything in this equation except for that value of lambda, which is the interleaflet coupling parameter. Yeah. Do we account for electrons?
And the potential bilayer, the potential domain of lipids, which also could have a polar group and would have an alternate.
That's a great question. Let me repeat it if you didn't hear. Have we accounted for the electrostatics in the system? Because glass is often charged. In our system, all of the different lipids that we're using, so our two different phospholipids have the same head group type. The two different phases have approximately, first order, the same amount of cholesterol in the two of them. So there should not be a differential effect. There may be a net effect, but that would not change the coupling parameter to my knowledge. That's a great question. Thank you. Are there others before I head on? Okay. And that gives us lambda equals 0.016 times 10 to the times 0.016 plus or minus 0.004 kT per nanometer squared. Yeah! It's so very warm in here, isn't it? Okay, so I can tell you are excited as I am about that result because we can take this measurement with no measurement before and now put in 0.016. Who is super excited about this experiment? My colleague, Michael Schick, which is really nice because I see him every week. It was kind of a relief to make sure that has actually turned out that way. You know, it didn't have to turn out that way. So physicists, we now know that physics works and we're very excited, but really this is a joint colloquium. Who cares? Well, MCD people, it all depends on how you like to think about cell membranes. If you like to think about cell membranes as containing some sort of nanodomain, some sort of region of different lipid composition, well, we know that cell membranes have different lipid compositions on the inner and the outer leaflet, on the cytoplasmic side and the outer leaflet of that cell membrane. So you can think of lots of interesting things happening. For example, the ligand binds to the outer leaflet of the membrane. How does that information, can that information be transmitted to the inner leaflet? Well, we're used to thinking about proteins doing all of that. Now, here's a case in which we're thinking about only the lipids. So think about what your favorite size for a domain in a cell membrane might be. If that domain is bigger than 10 nanometers, then just due to the lipids alone, there will be coupling between the two leaflets of that bilayer, which I think is not something that we're used to thinking about. Now, that number, the absolute magnitude of that number is going to change if we change the size of that domain, right? That number comes in kT per nanometer squared. So if you make a bigger domain, you're going to get bigger coupling. And it will also be the case that by tuning the lipid composition in the system to be, say, closer to a critical point in which the composition between the two different regions is less pronounced, then you should decrease that number. But what I really very much like is to be able to give an order of magnitude value to the community for thinking about how that coupling should persist. Good? Yes? What's really lovely is that when the question was, what temperature are we looking at? Is this body temperature? Is this room temperature? What's really lovely about calculating things in terms of kT is that we're using Kelvin, and there's not a big difference in that case. It's also the case that by changing the lipid composition, we'll change the temperature at which the system demixes. Okay. Now, I'd like to make a final sort of technical note. And perhaps we can end a little early and go out into the cool breeze, about thickness mismatch. When I drew all of my cartoons for you about this particular system, I drew it as if there was just one smooth sheet with no difference in the thicknesses between the two different phases. And that led us to think, the reason that we did that is because we didn't just want the shear flow above of that liquid to catch on the end of that domain and to act like a sail and to drive it forward, right? Our equations did not consider anything like that. So we chose our lipids on purpose to try and minimize any thickness difference between the two different phases. Now, what if you were trying to maximize the thickness difference? And why would you do that? Because perhaps you have different proteins that have different lengths of hydrophobic regions, and you'd like them to partition differently into the two different regions in a cell membrane. Okay, so this is work done by Joan Bleeker, Jonathan Litz, and again, Matt Blosser, in collaboration with some really wonderful AFM experts. Okay, so here was our thought. When we think about cell membranes, we usually think about an ordered phase as being thicker, and that's often observed. But when we get near a critical point, then those two different phases should be approximately the same thickness because they have approximately the same composition. Could we ever get a freak like this? Could we ever get a thicker liquid disordered phase than the ordered phase? Okay, well, if that's gonna work, what do we need? The difference between the two phases is predominantly in the types of lipids. One of them has a high melting temperature and one of them has a low melting temperature. So we would like the one that has the low melting temperature to be a long one, a long unsaturated lipid that we're going to couple with a short saturated lipid. And then Joan looked at this by AFM. We're going to use the same trick. Make vesicles, lower the temperature so they phase separate, put them down on a substrate, splat them, and now there's going to be a little thickness difference between the liquid ordered and the liquid disordered phase that we're going to see by AFM. And the short answer is, no. We have not yet been able to see a case in which we can get a thicker liquid disordered phase. I still think it should happen, but not yet with the lipids that we've chosen. So to make a long story short, yeah. Oh, that's a really good question. So what is the difference between the liquid ordered and the liquid disordered phase? Partially you can guess from their somewhat goofy names. If you do an NMR experiment to look at the chains of the different lipids, then the order parameter of the lipids that are in the liquid ordered phase, there's a higher order parameter in that case. Okay, right. So of all the different systems that Joan tried, it is either the case that the liquid ordered phase is thicker than the disordered, or that the membrane demixes below room temperature, so we can't do room temperature AFM, or there's no coexistence whatsoever. Very frustrating to me. I'm sure very frustrating to Joan too, because she did the work. So now that makes a question to you. If you're interested in a question like, is this at room temperature or is this at a physiological temperature, maybe we as a community would like to know how to choose our lipid systems in order to get a phase transition at an interesting biological temperature for our system. Okay, so how would we go about predicting what that is? How would we choose lipids to, say, for instance, maximize the miscibility transition temperature? Okay, so here's a test for you, because I know it's getting late and it's a very warm room. On the y-axis of everything is the highest miscibility transition temperature. What does that mean? That means that at a temperature above that temperature, the vesicle would be in one uniform phase. Below that temperature, there'd be two coexisting phases. Now, my challenge to you is to figure out which of these plots is going to give any correlation. Which x-axis is going to be predictive of what you see on the y-axis? Is a difference in the number of carbons in the tails on those lipids going to give you the highest melting temperature? Is it going to be a difference in thickness for the bilayers if you have a single type of lipid? Is it going to be an estimated thickness difference between the two different phases? Do you have a choice yet? Is it going to be the highest melting temperature of any lipid in the system? Or is it going to be a difference in the melting temperatures between the two different lipids? I'll give you, do you have a vote? Let's see, let's number them. Let's go 1, 2, 3, 4, 5. We have a 5, we have a 5, we have a, oh, 5, 2, 2, 2, 4, oh, awesome, none of them. It's none of them. It's none of them. Oh, it's none of them. It's none of them. So, bad news. Well, I don't know. Maybe that's a little bit judgmental, isn't it? News, I should say news. News, there is no clear trend to predict the highest melting temperature. Awesome news. Experimentalists still have worth in the world. We still have things to discover. I do not know why it is like this, and I would love to have some way of predicting that if I had the lipidomics of some bug, and I would know then what should be the mixing temperature for that membrane. I have no way to predict, which is good for my group. Okay. Moreover, the ratio, so this just tells you the particular lipids we use. This is the highest melting temperature in the system, but it does not tell you what ratio of lipids that highest temperature falls upon. What I'm trying to say is that if this is temperature going up, and this is all of composition space, the way that we tend to think about phase diagrams in this system is as if we have nested bowls in our kitchen. And we would think about little ants that are crawling upon the bowl, and the highest point of the bowl is always at the same location on our floor. But these are very different sized bowls. They're not necessarily hemispherical bowls, and they can be offset as well in a different way for each different system. Hooray, we still have really interesting work to do in the field. And with that, I need to thank a lot of people. I need to thank my current group. It's such a pleasure working with fantastic students. It's been such a pleasure talking to students here. There are some really talented alumni as well who are cited in this talk. There are collaborations. I cannot do this work without really wonderful collaborations with the theorists. So there are more here who I'm looking forward to more collaborations with in the future. And funding, if you reviewed my grant proposal, thank you very much. And I am here today because my co-director of Future Scientist Training Program is making sure that our daughter got to school on time today. There are a lot of things that enable us to be happy scientists. Obviously the great students, and obviously the great collaborators, and obviously the funding makes us happy. And there are other structural things in our lives that really enable us to do the fun science that we do. And I think we should acknowledge all of those things. And with that, I'd also like to acknowledge you. Thank you for the questions. I'd be happy to take more. Yeah, David. Uh-oh.
So what was the origin of the energetic penalty?
That's for you theorists to figure out. I have no idea. So I don't know if you heard his question in the back. What is the origin of this, this interfacial energy penalty? Like, why is it the case that if you have a mismatch, I can make up 3 reasons. And as I was trained as a physicist, and that's what I'm trained to do. So here we go. One could be that there is some interdigitation between the tails. How would you figure that out? Well, ideally what you would do is you would have lipids that have different lengths of tails so that they would interdigitate more. I would love to do that experiment. What I did not tell you is that we're already in the case for our experiments where we are at the maximum that we can measure. So we already chose lipids that have about the same length. If we're already at the case that we are pushing so hard that our microfluidic cell pops open, we have to make a huge, ugly clamp to keep the thing closed. Not only that, even if we can keep our microfluidic cell closed, if we push harder, say, I'm doing that annoying professor thing where I'm saying we, it's all Matt. Even if Matt pumps fluid through his microfluidic cell harder, the failure mode of this system is that the entire bilayer delaminates. You're like, look at that beautiful bilayer. Gone. So I would love to know some way to turn down the coupling between the two leaflets, and I cannot think of a good way to do it. If you have an idea, I would love to know. That would allow me to get at that mechanism. So one of those mechanisms is interdigitation. Another mechanism is the flip-flop of the cholesterol, which is supposed to happen on millisecond timescales. Another is that cholesterol, because it has this very tiny hydrophilic head group of just a hydroxyl group, is supposed to be bobbing up and down in the membrane, and that could make some sort of coupling, but I don't know. Yeah, Grant? Explain what you mean by mathematically. Where does the... So I think the way that Miko derived that is just to assume, let's say that there is a misregistration energy, free energy per unit area, which is not as satisfying. Doesn't get at the mechanism at all. Yeah. Oh, I'm sorry. And then there. Yes. Ah. That's a really great question. To what extent does one lipid molecule in one phase escape into the other phase? No problem. So you can label a lipid and watch it traverse the entire system. It's not as if there are grain boundaries in this system. Everything is fluid. The difference is just in the ratio of the lipid in the two different sides, and any individual lipid has the ability to traverse the entire system. No clue. I have no clue. It's so fun. So in the back first and then. Oh, you don't. Come on. Yes. Yes. That's right. Poof, you got it. All right, man.
So when you make this vesicles, you inject water. You inject water inside the bilayer to make vesicles.
Oh, so that's a good question. So we make them by electroformation, which is another way of saying black magic. Ooh. Oh. Yeah. Ah, we have done that experiment, but it wasn't in the past year, so I didn't tell you about it. So we have done experiments in which we have added charged lipids to our membranes, and the particular charged lipid that we're going to use is one that is the high melting temperature lipid, so it should partition, and it does. We showed that it partitions preferentially, highly preferentially, to one of those phases. Now, it should be the case that charges don't want to be next to each other, and so now if we add salt to the system, we should see a difference, should see a difference in the transition temperature, and we hardly do. And I don't understand why, but it made a very pretty paper. So, you know, it's a joy doing experiments, because often you think you know what's going to happen, and then often we're talking this morning about just playing around and the things that you find. It's really a pleasure of this job. Yes?
Are your instruments sufficiently spatially precise to be able to measure any compositional differences between the ordered and the disordered phases? For example, is the ordered phase enriched in cholesterol?
Ah, that's a great question. So the question came about is, are your experiments, is your instrumentation positionally precise enough to tell the difference in the composition? We don't need that positional. We can tell by mapping out the tie lines in the system. There are two different ways to do that. So do you speak tie lines? Okay, that's okay. To figure out what the composition of the two different phases is. Now, there are two different ways to do that. One is by fluorescence microscopy, to walk around on that triangle in that composition space and think about the different ratios, and to look at whether we have very tiny black domains on a white background, whether we have about half and half, whether we have very tiny white domains on a black background. If it were the case that the primary difference between the two phases were in the amount of cholesterol, then we would expect, as we, on the triangle that's moving up, that as we move up in that diagram, that we would go from little white domains to little black domains. That's not what we see. We see that transition the other direction. So it turns out that the primary difference between our two phases is in the type of phospholipid. That was an unexpected discovery. That was something that was really exciting in the field. Okay, in 2003, but but I still get excited about it because it was really not what we had expected. It was completely different than what you see in lipid monolayers. So that's a semi-quantitative way to do it. The quantitative, well, not the a quantitative way to do it is by NMR, and then to use a deuterated phospholipid. And now to look at the difference in the splitting, you can see coexistence in the two different phases. And now you can quantitatively map what the compositions are of the two different phases. And that says, again, that the primary difference between those two different phases is in the phospholipids, not in the amount of cholesterol. There is a slight difference in the amount of cholesterol. So it is true to say that one is cholesterol rich, but that's not the primary difference. Answer, okay. Yes, Kranti? Yes. So it's, the way to think about it is there's this fluid flow on top, and then it's the shear that the bilayer will experience. That's the shear that would happen across the length of that bilayer. Yeah. I mean, really what's happening is this, right? Are there others? Yeah.
In your gov, the ordered phase domains sort of use over time to giant loss.
Yes.
What keeps them in the ordered phase from doing that like
Oh, isn't that the question? So the question was, why in a plasma membrane don't you see these large-scale phase separation in the same way that you see it in the giant unilamellar vesicles in these model systems? Okay. There are many different answers to that, depending on which research group that you talk to. I have not studied it myself, so I'll just report to you what some of those are, and then tell you that there's a beautiful gold mine in the literature for you to explore further. So it is not the case that the difference is merely due to having many different kinds of lipids in the system. So if you take many different kinds of adherent cells, and are slightly mean to them so that they make a bleb, and that bleb comes off, that's called a giant plasma membrane vesicle. And now decrease the temperature of that giant plasma membrane vesicle, it does the same thing. First of all, it has critical phenomena. At the low temperature, those domains are huge. They coarsen to be the size of the vesicle itself. Now that system has a huge complexity of lipids. It has a huge complexity of proteins. It does not have the proteins that were associated with the cytoskeleton, because that's where it ripped off from. And it has evidence of not exactly 100% of the asymmetry between the two leaflets that were before. Now, given that, one hypothesis is that the reason that those domains are small in the cell system is because the underlying cytoskeleton is there, and that's creating a length scale that is limiting the coarsening in the whole system. So that's just one of several hypotheses. Yeah. What's the difference in size between the vesicles that we were playing with and a normal cell? Well, it depends on who's making the vesicles, and it depends on the experiment that we're doing. In some cases, we would like to get vesicles as large as possible. Experienced graduate students can make them 100 microns across, which is bigger than we think of a lot of cells. So most When I want to pull a number out of a hat and think about a size of a cell, I usually think about 10 microns. Is that about the size that you think of them as well? You guys are the experts, MCB. Okay. But we can also make vesicles of that size. It's just easier for us to make bigger ones. Or rather, it's easier for us to do experiments with big ones. Is that it? I'd be happy to take other questions out in the breeze if you'd like to ask more. Literally the melting point of the audience and the speaker. Thank you.